Mm. And so again, we're back with your activity 22. That is your lipids from the brain. Okay, and so accordingly, and so your lipid constituents of the brain, you have there your phospholipids, your glycolipids, together with your sphingolipids and the small amounts of triglyceride accordingly. And so of course, and so in this activity, and so you're going to do your uh we're going to extract lipids from your brain in a way. And so, of course, with that, and so our sample, main sample with this one is, of course, a brain, no? And so you have your pig's brain with you. Okay, and so, of course, and so we're going to extract, and so using your acetone, and then you have your, yes, also your acetone together with your ether. And so with that, okay, and so let's take a look at your activity 22, your lab. So moving on, you have here your Salkowski test. And so again, and so you have your Salkowski test. Okay, and so this one is a test for your cholesterol. And so of course with that, if you're going to test uh your pig's brain, and so with this one with your Salkowski test, and so remember that your pig's brain, and so it contains a lot of cholesterol, and so it has a concentrates of cholesterol present in the brain, and so that is why, and so your expected result for this one, your Salkowski test, should be positive. Okay, and so you should have a red color or probably purple color for this one. And so a red color. Okay, and so your Salkowski test, and so it utilizes your chloroform and your concentrated sulfuric acid again, no? And so with your chloroform as a dehydrating agent, and then of course you have there your red color in the upper layer, and so you have your yellow with a green fluorescence, what you call this, in the in the bottom layer, and so of course you have there your sulfuric acid still. Okay, and so that is for this, of course, and so your sample, and so you have your uh macerated macerated pig's brain here. Okay, and so that is your sample. Okay, and so your Salkowski test, and so moving on to your Lieberman your Lieberman-Burkhardt test. And so this one again is another test for your cholesterol. And so but this, and so it uses your cholesterol, your acetic anhydride, and your concentrated sulfuric acid still, no? And so the positive result, and so is a production of bluish green to emerald green color. Okay, and so with this again, and so you already know this one, your Lieberman-Burkhardt test. And so your acetic anhydride, and so is utilized as your dehydrating agent, and your sulfuric acid as the oxidizing agent, no? And so that is for your Lieberman Burkhardt again. And so this one, and so is a test for your cholesterol, and so in the presence of cholesterol in the sample, and so it will give you a bluish green to emerald green color. Okay, and so when you tested your what do they call this, when you tested your uh, pig's brain, and so with your Lieberman-Burger test, of course, it has cholesterol in it. And so just the same with your Salkowski, and so it will also give a positive test. And so giving you, and so a bluish green to emerald green. Okay, and so that is for your Lieberman-Burger test. Okay, and so moving on to your phosphate ions, no? And so you have here the test for your phospholipids, and so what are you testing here is the phosphate ions, no? And so with this, of course, you did is to macerated an amount of probably, I think that is 5 grams of your pig's brain, and then you added ether or chloroform, no? You added ether or chloroform, and so, and then of course you mix it thoroughly, and then you filter using a cheese cloth or yes, your filter paper or your cheesecloth, no? And so, but then what you used is a gauze, no? And so you have there, and so of course you did the filtration, and so and then you added your fusion mixture, and so your fusion mixture here is a mixture of 1 gram potassium nitrate and then 2 grams of your carbonate, no? And so and then of course you add and then you add it to the filtrate this one, and so they use this fusion mixture and then you heated, no? And so you heated in a flame, direct flame, no? And then after that, and so you added you added water, and then you what do you call this? You filter it again, and so you filtered it again, and so finilter nyo ulit, and then of course the filtrate, and so you take the filtrate, and then from that filtrate, and so in the filtrate, and so you added a drops of ammonium molybdate solution. And so if you recall, and so previously you did this one, and so in your bile, what else? You have actually others, even in saliva. And so you did this, okay? And so with this, and so this is actually a test for your phosphate ions. You added ammonium molybdate solution, and so of course your sample your reagent is your ammonium molybdate solution. And so with this, of course, and so in the presence of phosphate ions, and will give you a bright yellow precipitate. Okay, and so we'll give you a bright yellow precipitate for this. And so that is for your phosphate ions. You are not testing for the fatty acids here. You're not testing for lipids or glycerol in here, but then you're testing for your phosphate. Okay? And so you did this actually previously. Okay? And so huwag kayong malito. Okay, and so you have there, and so the production of bright yellow precipitate. And so if there is a bright yellow, then meaning in your sample you have your phosphate ions. Okay, and so moving on to your test for your glycolipids. Okay, and so your glycolipid, and so that is a combination of your your glycolipid, that is a combination of your lipid and your carbohydrate. And so with this, what are you testing here is your your carbohydrate portion, okay? And so in your glycolipid test, no? And so you have your sample, you have your test, and so for your glycolipid, and so you have your anthrone test and your Molish test. And so, but then other agent that was given to us in the laboratory was your Molish test. Okay, and so it was the Molish test that you did. And so, but then, and so let's take a look at the two general tests for carbohydrates. Okay, and so you have here your of course, this one general test for your carbohydrates, and so either combined, either just a carbohydrate or combined with other compounds. Okay, and so we did this one previously, and so before, and so in your carbohydrates, and so yes, and so you already know the principle behind this one, and so in the positive result for this. Okay, and so this one, and so a rapid detection again for your carbohydrates in sample. And so again, and so either they are combined or free. Okay, and so with this, of course, your sulfuric acid will dehydrate the monosaccharide. Sorry, it will dehydrate your carbohydrate, and so to form your furfural derivatives. Okay, and so in this furfural derivatives will interact with your anthrone, and so it will interact with your anthrone to give you, of course, a blue or green color with this one. And so your reagent that is used, and so you have here your uh it will what you call this, your reagents use, you have here your anthrone reagent and your concentrated sulfuric acid. Okay, and so that is for that one. Okay, but then of course first you prepared, and so before you did your Molish test, and so you prepared first your sample. Okay, and so that is you have your ethanol, and so in a boiling water you place a an amount of ethanol in a boiling water bath, and then of course after that, and so you have this an amount of brain, and so and then you macerated it. Okay, and so you macerated it, and then you what they call they say, uh you add the hot alcohol, no? Ayan, so you added the hot alcohol. And then after that, ayan, so you filtered. And then you added your 10 drops of your molish and then your anthron test. Of course, and so with it, being it has your glycolipids present, then you have here, and so it will give you a positive test for it. Okay, and so you have there is a positive test for your Anthrone is a production of your blue or green color. Okay, and so that is for that. Okay, moving on. And so you have your Molish test. And so this one is what you performed in the laboratory for your glycolipid test under your lipids from the brain experiment. Okay, and so your uh okay, and so of course all carbohydrates should give a positive result for this one. Okay, and so you have here your Molish test, and so it uses your okay, and so it utilizes your 10% alpha naphthol and in ethyl alcohol, no, and of course you have there your sulfuric acid, concentrated sulfuric acid still. Okay, and so your sulfuric acid, and so that is your dehydrating agent, and so to produce your furfural derivatives, and then your what they call this, and then your positive result is your violet ring at the junction. Of course, this one, and so your furfural will combine with your alpha naft with the naphthol. Okay, and so to give you that color. Okay, and so you have there, of course, again, your Molish test, and so it's a general test for carbohydrates, and so either combined or not. And so again, your glycolipid, it has carbohydrate in it, and so with this one, of course, and so with this, of course, and so the carbohydrate content, and so is responsible for the positive result. Okay, and so you have here, and so again, your Molish test, this one is a general test. Okay, and so it indicates that there is a carbohydrate in a solution. Okay, and so that is it for your Molish test. Okay, and so moving on. And so you have here your uh, s- uh, solvents that can uh, extract your cholesterol, and so you're from the brain. Okay, and so that can extract your cholesterol, and so of course you have here your relatively the selection of your appropriate solvent, and so is the most critical factor in extraction of lipids accordingly. No, and so uh accordingly, and so your lipids will what you call this, they are soluble in nonpolar organic solvents, no, and so but of course, and so your polar lipids accordingly, and so are dissolved in polar solvents, and so with this, and so you have here to extract accordingly efficiently extract your lipids, and so from a biological tissue, and so you need a mixture accordingly of your polar, and so polar solvent, and so that is to disintegrate the lipids from cell membranes. Okay, and cell to disintegrate the lipids from cell membranes, and your nonpolar, and so to dissolve the lipids in to dissolve the lipids. Okay, and so with this, accordingly, your chloroform and your methanol, and so that is a what you call this, the commonly used solvent, no, and so to extract lipids uh from human tissue, no, ayan, so at actually a 2:1, two parts of chloroform and one part of methanol, no, and so you have here for your examples, you have your chloroform and methanol. Ayan. You have your hexane and acetone, your ethyl acetate and ethanol. Ayan. So these are actually the most commonly one. And so, but then your most common is your chloroform and methanol. Okay, and so and the rest, and so you have your hexane and acetone, your ethyl acetate and ethanol, butanol and methanol, your butanol, methanol, and chloroform, your ethanol, and so your acetone, your ether, and so those items, and so they can also be used to extract. And so let's take a look. And so your hexane, and so this one can also uh, extract lipids and fat-soluble vitamins. Your diethyl ether, and so you have this one is for general purpose extractions accordingly. Your diethyl ether. Your ethanol, and so steel, and so for polar and non-polar substances, and then your methanol, and so also, and so for extraction still your chloroform, and so this one is what you call this for extracting lipids and other non-polar compounds. Okay, and so those items, and so the most common one, and so you have your mixture of chloroform and ethanol. Okay, and so moving on, you have, let's take a look at your phospholipids, and I mean at your lipid classes that are actually present in the brain. And so you have here, you have your, of course, these lipids that are present in the brain, and so functions for what we call this structure of the cell membrane, and so also signal transduction, and of course, maintenance of your myelin sheath. Okay, and so others, and so it contributes, no, and so sorry, and so they contribute for synaptic function, and then also neurotransmission, and then of course for the health of your nervous tissue. Okay, and so let's take a look at this ones. And so you already know this, and so you have here your phospholipids, your glycolipids, and your sphingolipids. Okay, and so other than that, and so cholesterol is accordingly part of this one. Okay, and so your phospholipids, you have there your phosphatidylcholine, your phosphatidylethanolamine, your phosphatidylserine, and the rest. Okay, and so your phospholipids, ayan, so they are the major component of your cell membranes accordingly. Okay, and so of course with that your cell membranes, and so they are also present in the brain, no? And so you have there. Okay, and so other than that, you have your cholesterol. Your cholesterol, and so this one is an essential, accordingly, component of your cell membranes, no? And so that is, it plays a role in what we call this, uh, in myelin sheath still, and of course, maintaining your membrane fluidity. Okay, and so, and then of course, you have and so your glycolipids, like your glycolipids, which include of course your cerebrosides, no, and so you have there, and so of course your glycolipids, this one contains carbohydrate residues, no, and so you have here, let's take a look, you have your cerebrosides, and so, and then other than that, you have your, yes, you have there your cerebrosides. Okay, and so you have there, and so as what you call this uh component, I mean, as a representative of your glycolipids. And then you have your sphingolipids, and so they are what they call they are actually this sphingolipids accordingly are relatively part of somewhat part of your glycolipid, but then or the other way around. Okay, and so you have here, and so uh they are structural components still, no, and so and uh also, and so they insulate your nerve fibers. Okay, and so that is for your uh phospholipids, and then your glycolipids, sphingolipids, and your cholesterol. Okay, moving on. Okay, and so let's take a look at your sterols, of course. And so your sterols, and so these are actually under your sterols, and so they include your aromatic alcohols. And so these aromatic alcohols, they are actually derivatives of cyclopentanoperhydrophenanthrin. Okay, and so or you have it as your steroid nucleus. Of course, common to all your sterols, you have your cyclopentanoperhydrophenanthrin ring. Okay, and so this one is your cyclopentanoperhydrophenanthrin ring. This one is your steroid nucleus. Okay, and so your steroid nucleus is a combination of your cyclopentane and your perhydrophenanthrin ring. Okay, and so this one actually, and so on your left on your right side, and so on the right side, this one is an illustration of your uh cyclopentano perhydrophenanthrin ring. Okay, no? And so you already have your cyclopentane and then your what they call this perhydrophenanthrin ring here. And so you have there. Okay, and so still cyclopentane, you have it as the five carbon atoms. And so cyclic five carbon atoms, no? And then on the right, on the left side, and so you have here your phenanthrin ring. And so you have it as your phenanthrin ring. Okay, and so that is made up of three benzene rings. Okay, and so three benzene rings. Okay, and so in this one, and so there's a phenanthrin ring made up of three benzene rings, and so meaning they are unsaturated, no? And so how is it converted into your perhydrophenanthrin? And so this one is and so in this case, and so when there is an addition of hydrogen that is attached to each carbon atoms of your phenanthrin ring, then that is and so it will form your perhydrophenanthrin, no? And so of course there is a disappearance of the double bonds already in this case, no? And so with this, and so dapat supposed to be CH lang ang nandito, no? And so you have it as CH only. But then after that one, of course, when you added additional, and so when you added additional hydrogen to each carbon atoms, naging CH2 na siya. CH2, and so CH2, 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 CH2. Okay? Ayan, so that is for that. And then, this perhydrophenanthrin ring will fuse with your cyclopentane, giving you that steroid nucleus. Okay? And this one is common to all of your sterols. Your cholesterol. Ayan. So your cholesterol, and so this one is the representative of your sterols. And so this one actually is the most abundant. No, and so this one is widely distributed in the body, and so in all cells. No, and so uh uh a significant amount is not significant, and so a concentrated amount is present in the nervous tissue. Okay, and so this one, and so is the structure or your structural formula, and so your structure of your cholesterol. Okay, and so that is your structure of your cholesterol. And so with that, okay. And so there is actually a hydroxyl group that is present at your position 3. And so that is at your carbon 3, 1, 2, 3. Okay, and so at your carbon 3. And then of course there is a what you call this double bond at your 5 and 6. No, and so between your 5 and 6 carbon atoms. No, and so you already have a double bond here. Okay, and so your cholesterol. And so this one is a lipid molecule, and so of course it plays a role in maintaining your cell membrane integrity still, okay? And a precursor for your steroid hormones like your testosterone, no? Okay, and so and other than that, and so it contributes for, I mean, this one is the source of your bile acids also. Okay, and so of course your this bile acids, and so they help in the absorption of your fats. Okay, and so you have here they also serve as an insulator by forming part of your myelin sheath. Okay, and so that is for your cholesterol. And so and then of course this one is your formula. The structural formula is you have a C27, you have 27 of carbon atoms present, and then you have your 46 hydrogen, H hydrogen, I mean H46 and 1 oxygen. And so 1 oxygen in your hydroxyl group. And so giving you C27, H46, O. Iisa lang yon. Okay, and so that is for your cholesterol. Okay, next, and so you have your phospholipids. And so with your phospholipids, and so you have there your lecithin, and so your cephalin, and then you have your lipositol, your phosphatidylserine. And so with this, please, uh, with this ones, and so please refer, and so to your, to the importance of this one in the other uh videos. Okay, and so with that, your lecithin, and so with this, of course, and so the what you call this, and so they have roles in maintaining of your structural integrity still, and so you have there, and so precursor of your neurotransmitters, and so your lecithin, and then you have your cephalin, and so this one is what they call this a thromboplastic substance. Of course, you have your cholamine, ayan. I mean your ethanolamine, your what they call this, yes, your ethanolamine, and then you have your lipositol, and then you have your phosphatidylserine. Okay, and so your phosphatidylserine, and then your phosphatidylinositol. And so please uh, refer to the videos, to the other videos regarding this importance of this function. No, okay. And then let's take a look at your polarity of your phospholipids. And so how does it, what you call this, affect? And so it's the lipid bilayer. I mean the cell membrane. Okay. And so with that, and so it functions, of course, it forms part of your cell membrane. Okay. And so of course your cell membrane that is made up of your lipid, you have your lipid bilayer in that, and that is amphipathic. Amphipathic in nature, no? Ayan, so you have there, and so you have there your amphipathic nature of your phospholipids. With that, you have your hydrophobic and your hydrophilic regions, no? And so your hydrophilic or the phosphate, your hydrophilic heads, and so this one forms, this one is your phosphate, and so uh what you call this, are in the outer area. And so note that previously, and so when this one, when your phospholipids are suspended in water, and so they spontaneously arrange themselves, no, and so in water, and so giving you your hydrophilic heads or your phosphate part, and so is in the outer area, while your fatty acid tails or your hydrophobic fatty acid tails, they are in the inner area. Okay, and so with this, of course, and so it separates the inner and outer environment of your cell. Okay, and so note that inside your cell, you have there your cytosol, and that is liquid, that is water. And so outside of your cell, you also have your water present. Okay, and so that is why, and so it will separate your internal and external environment. Okay, and so your, of course, with this, and so and that, and so giving you, separates your internal and external environment, and so it also functions for like semi-permeable, selective permeability. And so it will what you call this, allow the allow your non-polar substances to pass through. Ayan. Okay. And then of course, and so its functions, I mean it's what you call this, for its function still in the polarity, and so functions of this one, the polarity that the polarity of your phospholipid also affects, and so your, what you call this, the fluidity of your uh the fluidity of your cell membrane, no? And so with this, of course, and sabi natin last time that in your degree of, I mean that you're unsaturated, and so they do not form a rigid they do not closely pack. And so meaning they are more fluid, no? And so hindi sila rigid. And so kapag if they will not form a closely packed structure, then they are not rigid, no? And so they are still fluid, no? And so they can move freely. And so with that, and so your degree of unsaturation present in the fatty acid chains, and so will also influence fluidity. Remember that your phospholipids, you have your lipids present, and so you have there your fatty acids and that. Okay, and so the degree of unsaturation present, and so will influence fluidity. Okay, And so with that also, this fluidity will also allow proteins to move laterally. And then you have here, and so of course your the polarity of your phospholipids provide environment where the membrane proteins can be embedded. Okay, and so you have your membrane proteins can be embedded, and so you have there accordingly, and so there are parts of uh, your lipid bilayer, and so that is made up of your phospholipids, and so provides an environment, and so where your proteins can be embedded, and so you have your integral proteins, and so where can they can be embedded, no, and so and then of course with that, and so it allows them to perform their functions. Okay, and so that is for your activity 22. Okay, and so that is for your activity 22.